I think we're going to go ahead and get started. Thank you, everyone, for coming to our omics seminar. Today, we're doing an introduction on single-cell ATAC-seq, presented by Dr. Lindsay LaFave. I'd like to give her a quick introduction. Dr. Lindsay LaFave is a lung cancer biologist who studies non-genetic mechanisms of cancer progression. Lindsay completed her bachelor's in biochemistry at the University of Michigan in 2009. She received her PhD in cancer biology at Gerstner Sloan Kettering Graduate School in 2015. Lindsay is then trained as Damon Runyon Postdoc Fellow in the labs of Dr. Tyler Jacks and Dr. Jason Buenstrow at MIT and Harvard University. Lindsay is currently an assistant professor in the Department of Cell Biology at Albert Einstein College of Medicine and a member of the Montefiore Einstein Comprehensive Cancer Center. She has received several awards, including the NCI K22 Award, the inaugural AACR Career Development Award in Lung Cancer Research, V Scholar Award, and among others. And before we get started, if you could go ahead and go to our omics website, there's a link on the email that I previously sent out to all the RSVPs. There's an updated HTML file that you'll need to follow along the vignette. And with that, I'll let Dr. LaFave go ahead.
Thank you for that kind introduction. I'm excited to be here today to talk to you about single-cell ATAC sequencing. So I'll start off with a bit of an introduction about ATAC-seq and single-cell ATAC-seq. But today, the main thing I want you to take away from my part of the talk is what you can actually learn from ATAC sequencing and why that might help your research. So, and then after Anthony, the real expert, is going to walk you through a tutorial of how to actually analyze some single-cell ATAC sequencing data with a vignette. So as you all know, chromatin is very important for gene regulation in cells. It's important for the right genes to be turned on at the right time and turned off at the right time as well. And there's many assays along, I don't know if you can change to the laser pointer, sorry. So there's many technologies that you can look at different aspects of chromatin biology, but the main approach that has been applied for single cell methodologies is thinking about chromatin more generally. So thinking about open regions of DNA and genes that are not in the heterochromatic region, getting that information from sequencing, and then learning what we can from that information. So the main key point is that the regulatory regions of the genome are nucleosome free, and those open regions are then available and accessible to enzymes that can insert important barcodes as well as regulatory information, or let's learn regulatory information. So the way that we think about DNA, again, another aspect, rather than just seeing if its DNA is open or closed, accessible or inaccessible, what that is it provides a landscape, a permissible landscape for gene regulation to occur, where transcription factors can bind and either open or close chromatin. So sometimes the transcription factors are active players in the remodeling process, and sometimes they just make use of the DNA landscape that has been set for them. So ATAC sequencing is a really powerful tool to be able to understand these open regions of chromatin and then also get further information about transcription factors that may be bound there. So I'm saying may, and that this is not a direct assessment of gene expression. This is not a direct assessment of something that is actively bound to chromatin, but it's a way for us to make inferences and hypotheses about what actually may be at play. So the way this approach works, I think that there was a bulk analysis session already, so you might already be familiar with this. But the very basic premise is that we have this enzyme called TN5 transposase, which can insert into open regions of chromatin, but it's blocked from closed regions of chromatin. And then this enzyme has loaded adapter molecules, which are inserted across the genome. What's important to note is that the standard way people do these assays is that they buy this enzyme preloaded with adapter molecules, but I'll just briefly mention that these approaches can can be further extended to use multiplexed barcoding, and either, you can buy these loaded in a different way, or most people load them themselves to actually expand the barcoding space that they can use for experiments. But simply what happens then is that you get insertion of these adapter molecules, and then you can read those out by next generation sequencing. So very simply, you get this, the DNA sort of chopped up, cut up, as shown here by scissors, if you can see, and then you get these tracks across the genome. So what you end up getting is a read, or an individual fragment, gets mapped to the genome, whatever your reference genome is, and they assemble into these peaks, which are the key feature that you use to assess your data. So I think people are more familiar with RNA sequencing, and so typically transcripts are corresponding to expression of a gene. What's important to note is that in these tracks, you get rich information about gene regulation, where these peaks pile up at different regions. So at the promoter of a gene, the enhancer of a gene, and they can actually be relatively far away from the gene. So ATAC sequencing alone can't tell you which gene this peak is regulating, but we can make inferences, and this course I don't think we'll touch much on multi-omics, but now there's ways to combine approaches to kind of sort out some of these questions. So just the main takeaway from this assay is that you get these accumulation of reads in peaks, and then each one of these fragments are associated with a TN5 fragmentation event. So just densely, the way this works is that you have the insertion of these enzymes, and remember that there's also transcription factors, and not histone molecules, what the DNA is wrapped around, is not the only thing blocking accessibility to the chromatin. There's also the binding of transcription factors across the genome. We're not going to talk much about it today, but chromatin accessibility can also be used to predict where transcription factors are blocking the insertion of these adapter molecules. But the most important thing is, again, that information that we're gaining is trying to determine potentially where transcription factors are regulating genes. And we get these sorts of peaks, which we think correlate with gene expression. Honestly, many times they do, but they don't always, because we know there's many complexities to gene regulation. I just put this slide in here just to mention that the way this works is that when you have insertion of the enzyme, you have this sort of homology region from the TN5, which then you can use to amplify with the barcodes that you're adding. It's a pretty standard library preparation approach that was then extended to chromatin biology. And the way that I'm talking about this more generally, but we'll get into single cell in a moment. But the most important thing to also note is that your data is fragmented. And so if you run something like a gel or a bioanalyzer, what you will see is you'll get a fragmentation pattern. And this fragmentation pattern depends on the ratio of DNA to enzyme. So if you have a lot of enzyme, you're going to have smaller fragments. And a lot of times that happens with single cell biology, because we're working with single cells. And so many times, you do end up with a skewed ratio. But basically, you get this many smaller fragments and then this periodic pattern, which is basically the distance between a single histone molecule. And so you can see how that builds out periodically. And people that if they're more interested in doing these footprinting analyses, sometimes they scale down the amount of enzyme that they're adding so that they can get longer fragments. And so, yeah, these peaks generally on average are about 200 base pairs, but this is just sort of an average of the many peaks. So the thing with ATAC sequencing is you get a ton of peaks across the genome, and deciphering information from that is what we're going to talk about today. And again, important to note that the relevant peaks that change many times are not at the gene itself, but at some peaks that are proximal or distal to the gene. And I just threw in a slide here from the literature showing that you can have similar expression of a gene, like in this case, in this leukemia example of TET2. But if you then go and look at the ATAC sequencing data, the enhancer regions look quite different. So that's sort of the complexity that we're working with these assays in single cell biology, is that even though you may have a gene that has similar expression levels, when you go and look at the attack sequencing data, it might actually give you some new information that you wouldn't have by just having expression of a gene. So single cell attack sequencing. I first am just going to mention a little bit about the sorts of data that you get back, and then we'll talk about the approach itself. So you're again doing the same assay for single cell attack sequencing, but you're either using a droplet-based approach like the 10x controller, or another sort of split approach where you're multiplex barcoding with small pools of cells. But what at the end of the day is you're doing that attack sequencing strategy on individual cells. So what our goal is from the information that we get back from those inserted sequenced, the regions that are sequenced off of those adapter molecules, is to get fragments. So fragments from all across the genome, which are kind of the features. And then what we want to then do is identify some meaningful information from that sparse information. So the way that many of the standard computational tools put the data, like a way you, I guess, organize your data, and Anthony's going to go in more depth about this than I am, is you're working with large scale matrices. And so these matrices are basically a cell which has a peak and then fragment counts. And if you look at the numbers, again, it's a little tough to see up here, but either 1, 2, or 3. So like the various, or many zeros as well, which you have to deal with in the data set as well. But the main working data set is this matrix of cell by peak information. And then typically there's sort of a downsampling approach, and then to make the data more easy to work with. And then you get information where you can do things like sum this information in fragments across known transcription factor motifs. And then you can also do some similar analyses doing it on background peaks where you wouldn't expect to maybe see a change. And then the sorts of data that you get back from this are called things such as deviation or variability scores. So these are basically taking into account the fragments you're getting from transcription factors and then normalized to a background signal across the genome. One of the standard ways, the computational tools that people have started to develop, there's a number of these now, so if you go into the literature, you can find many of these that have slight pros and cons to them. But one of the first ones was this approach called ChromeVar, which is this tool that allows you to work with sparse chromatin data. But it's sort of the same idea. So you, as I mentioned before, so you have these regions or peaks, which we could maybe say are an enhancer or regulatory unit, as a matrix with individual cells. You can also build new matrices, similar to what I explained before, where you're looking at this with aggregated information in motifs. And you could also, so motifs typically come, they're these position weight matrices, which say, based on either some in silico or in vivo or in vitro data, where transcription factors are bound across the genome. And so you can use that information to then say, is there a fragment in said motif or not? You can also do this with something called k-mers. So k-mers means that you're going in not knowing what the motifs are, but sort of setting a number of base pairs that you're interested in seeing if there's some signature there. But basically what we're doing is then getting these matrices and then using them for downstream analyses such as cell type identification or looking at different motifs, which I would argue that's one of the most powerful things about single cell attack sequencing data, is that instead of looking at individual gene signatures, you actually get to look at gene programs that are changing. So transcription factors that are regulating a number of genes across the genome. And that actually helps with statistics too, because instead of just looking at a gene compared to a gene, you're looking at a number of genes across the genome at that motif. So I'm not going to go through too much of the model or the tool we're going to talk about today, because Anthony is all set to show you that later. But ArchR is another tool that has become a common tool in the field, which was developed by the Greenleaf Lab, one of the developers of attack sequencing. And this is sort of a nice approach. We like this one because one thing that it does is it can store your file in a unique file format, which Anthony will describe, called an arrow file, which allows you to kind of smoothly work within this tool to do a number of different analyses. But there is obviously caveats because when you move between these approaches, not every system uses an arrow file. But for the standard analyses that you're doing, ArchR is a really great program to be able to do some of the standard analyses we'll talk about today. That includes just looking at your data set and seeing if it's of high quality or not, and which cells that you should move forward with analysis. That also includes just doing general clustering. Here, this is a UMAP, which probably many of you are familiar with, but doing dimensionality reduction analyses with the matrices described before, and then identifying cell populations. And this tool has a number of different ways that you can try to sort out what's going on in your data set. There's also the identification of specific marker genes and different approaches. This is something I really want you to take home from ATAC sequencing data today, is that there are ways that we can infer the activity and potential predicted expression of a gene based on ATAC sequencing data. And so this is really powerful because what it allows us to actually do many times is move between tools that are used for ATAC sequencing as well as for RNA sequencing. Because what you end up with is a similar matrix of information about gene activity, variable changes at the chromatin level. And these tools are very powerful when you have variation. So when you're seeing that chromatin is changing. And this can also do these experiments we talked about, like transcription factor footprinting. And we won't get into this today, but if you have RNA data, you can also pair together ATAC sequencing and RNA sequencing data in this tool. There's a number of other tools, though, I would recommend if you're interested in this space to investigate, because they have unique features, and if you can move between them, that can be somewhat powerful as well. I also just wanted to mention this briefly. Those standard analyses don't take into one caveat, which is that TN5, the enzyme you're using, is known to have an insertion bias. And there are ways to deal with this, and ArchR has ways to also deal with this, but not in their upfront analyses. So it's something to keep in mind, in that there is some bias going into your analysis about where TN5 is going. And some tools have found some ways to get around this, but there's new papers that also suggest there's some complexities. And I recommend you check out the ArchR tutorial if this is something that you're interested in. So with ATAC sequencing, there's a number of different approaches that are out there now. If you go on bioRxiv, you can find a new approach all the time. The most standard approach is on this image here at the bottom, where you're using the 10x platform encapsulating a single cell and then performing chromatin profiling. The first approach was doing the same sort of idea with fluidics on the Fluidigm platform. And then I would also, if you're interested in doing things at scale, many of the atlases that have been generated, especially from Jay Shanduri's group, have been used with combinatorial indexing, meaning that you get to do multiple rounds of barcoding and then load these on to either an instrument or do a plate-based analysis. And what this does is, instead of the 10x platform gives you about 10,000 cells on a lane, you can definitely scale that up by overloading the instrument if you barcode up front. There's also a number of other approaches that have integrated ATAC, to do perturb ATAC, to do profiling of immune cells, et cetera. So I would recommend, if you're interested, to check those out. But the main thing that many studies report when they're doing single cell analyses is sequence-specific motif enrichment. And I'm just showing you an example of one of the first single cell ATAC sequencing papers from Jason Buenrostro's group. But what you're then able to do is look at variation in a data set. So you have a motif for a transcription factor, in this case, the Jun transcription factor. And then what you're able to do is look at the variability in some context. In this case, it's treatment versus control of this specific cell cycle inhibitor. And then you can rank the different factors or motifs that you're looking at and find the ones that are the most variable in your data set. And that means the ones that are changing the most. And so those are the ones that typically will have a very robust signal that you can then go after and further analyze. And then you'll commonly see these sorts of large scale clustering analyses. In this case, these are single cells. And you can then look at how these cells are similar or different from each other based on their chromatin accessibility across those individual cells. And the main way readout of these data, here it's shown as a heat map, can be shown plotted on a UMAP, can be shown many different ways, is looking at those aggregate, that aggregate chromatin accessibility data and looking where there's changes at sequence-specific motifs. And you can see here examples of many of them that are changing in this data set. So I'm just going to give you an example of how we've used this approach and how we can go back and forth between using single cell attack sequencing, learning biology, and going back to the data set. So this is an example of a combinatorial approach that we used. In this case, we use the TN5 barcode up front, but we load them, each pool of cells, with a different barcode. So instead of starting with the standard Illumina barcode, each of these pools have their own barcode. We mix them together, we split them, and then we do PCR amplification. So you do this with math. The math is very important, so that you, at the end of the protocol, expect that you only have, most of your cells have unique barcodes, 4 unique barcodes at the end. There's several companies now that have been working on commercializing this. So we used to have to do all of this in the lab, but there are now kits available that allow you to do the combinatorial indexing and also move on to the 10x machine so that you can really scale your experiments. So what we're interested in is thinking about how gene regulation in different cell types, especially in cancer, how that changes across diverse contexts and tissues, and thinking about that through lineage factors, where lineage factors are normally bound and how those programs change when these cells become altered. And we know that this happens at a number of stages across cancer progression, including initiation, through proliferation, progression, and metastasis. And I would argue that single-cell RNA-seq and now attack sequencing have shown the immense complexity of these sorts of diseases. And I'll show you some take-homes that we have that has been kind of a common theme across these diseases. But just as just an example of how to use this, we used this approach previously in a mouse model of lung adenocarcinoma, in which you can specifically engineer the KRAS mutation and P53 loss in a cell of origin. They're called AT2 cells in lung cells to initiate tumors. And what was interesting about this model is it goes through the same progression of human lung adenocarcinomas, but the literature has shown that there's not many mutations that are associated with these transitions. So for us, we thought this was a great platform to study why we're getting so much variation in these tumors when there aren't clear genetic variants that are explaining it. So we did an experiment where we took normal lung cells. I think it's always helpful if you have the normal cells, either in your data set or in someone else's data set, to help orient yourself, especially for cells that are new cells. So like you don't know much about them yet. In this case, that was our case with the cancer cells, but we did have some idea of what's happening with normal cells. And then we did that barcoding reaction that I talked about. And then at the end of this experiment, we ended up with about 17,000 cells that we used for profiling. Anthony will talk a little bit about this, but maybe not show all of the figures, but some of the main quality control things that you need to look at for attack sequencing data include something called the transcription factor, TSS, or transcription factor start score. So this basically is giving you information about chromatin accessibility at the transcription start site. You would expect, if you have data that's clean and not incredibly noisy, that many of your reads are going to pile up around transcription factor start site. So what this basically does is gives you that information about, do you have data that's just not noisy across the genome but has some logic? And then many people have a score. In this case, I'm showing one example where you can see this upper library maybe has the highest quality score, and that decreases in this experiment. There's also something called fragments of reads and peaks. So this is something, again, where you're looking at the percentage of fragments and peaks in your experiment. A peak, again, those predicted regulatory units. And I'm showing you this plot as an example because this is sort of a standard cutoff for what you would use for FRIP. And in this experiment, you can see that there were some cells that didn't quite hit that threshold And so they were excluded from the experiment. And again, you always get nucleosome patterning, so it's also helpful to look at these patterns and make sure that they make sense. Another sort of quality control you might see is something called a species mixing experiment, especially for these combinatorial experiments. So if you are starting something for the first time and you want to make sure you have single cell data, it can be helpful to mix cells from two different species together, in this case, human and mouse, and then make sure that at the end of your protocol, you're mostly getting cells that have information from just human cells or just mouse cells. And then again, this is the sort of data that you see, this peak-based information, which looks very similar. In this case, I'm showing you, we were able to profile 44 samples. This is all included in an aggregate track here. But what's helpful is that the bulk tumors look very similar to the single cell tumors, but you get this sparse information, those fragments across individual cells. And you can also look in this case, we had a deleted region, and you could see that that was missing from ATAC sequencing data. So again, Anthony is going to walk you through how to process these data, how to do these sorts of dimensionality reduction analyses. UMAP is sort of the commonality in the field. It's a 2D representation or visualization of your data. It's obviously not perfect, so you really should use this mostly to make hypotheses and think about generally similarities between cells within representative clusters. But again, it's a 2D representation of varied heterogeneous, complex data. So in this case, we had the normal cells, and the main take-home point here was that the tumor cells were very complex compared to those normal cell populations. But the challenge with working with this, especially when the field was somewhat new, was that it was a continuum of states. It's not that the cells were going from one state to another, it's this complexity within the data that you could potentially get back. And so that's what these sorts of newer computational tools, using motifs, using gene score, or activity scores, which we'll talk about in a moment, can help infer more information here. But generally, we saw from the initiating cell population, this alveolar type 2 or AT2 cell population, the cells became very complex, but across the continuum. And then we found that they sort of jumped towards this distinct cluster of cells, which we think represented the transition from epithelial to mesenchymal transition. And I'll get back to this point, but we could actually find that there were some cells in here gray, which we found from the primary tumor, that we could learn something about from single cell ATAC sequencing data, even though they looked somewhat similar to other cell types. And what you can also do is you can go back, if you do these sorts of barcoding approaches, you can kind of aggregate data and have samples within the data set, and then find that in our case, we found these sort of continuous changes in tumors that were across individual isolated tumors. So what are the sorts of data now that you have an experiment and you've found that you have some differences? What's the sort of things that approaches that you can use to get some information about the gene regulation in these cells? So one example we've already talked a bit about is a motif score. So looking at the openness of motifs across the genome. This concept is the one that might be a little bit newer, this idea of a gene score. So a gene score is looking again at the transcription start site and looking at the chromatin accessibility around this gene to make inferences of changes in variability at that specific gene. So you can then look across all the genes in the genome and get these scores. So when we did this in the first iterations, it was really just an exponential decay function away from the start site, but there's challenges associated with that. There are genes that are, sorry, I don't have it here, but I have it later, I'll show you later. There can be cells adjacent. So in the R tutorial that Anthony is gonna walk you through, they've actually iterated a little bit to block adjacent genes and to also change that function a little bit as well. But this is very powerful to be able to gain information and correlative changes, genome-wide motif changes to changes at individual loci. And so from these data, we were able to do these sorts of analyses. So first I'm showing you the aggregated motif information. So essentially we had in this data set 12,000 tumor cells. They were clustered based on their similarity of motif, but you start to see red and blue. So a lot of variation at the individual motifs. But again, this is very dense data. So thinking about how this is meaningful to biology can be a little bit challenging at first glance. So one way that we can do this, I pulled this example from the ArchR tutorial, but one thing that we can start to do is look at motifs that are changing across the genome, and what are the most significant ones, what are the most variable ones in our data set. So there are data sets like scBP in which these position weight matrices are used to infer information. But what you can do is you can plot things where you're essentially looking at either in cells, but you can also, once you know what your cell populations are, you can kind of aggregate that information and look at differences between populations. So one point I want to make is, in this case, in this plot, you have this heat map with a motif and yeah. Oh. Okay. All right, we're not, yeah, we're not advancing, it seems. So I could.
Maybe just like stop sharing for a second.
All right. Oh, it is now? Okay, we must have just had a lag. Okay. So you get these sorts of motifs, and then you have these, you can generate heat maps, which we'll walk through later, but these are basically in a data set, some specific motifs, and then in this case, they're looking at populations and looking at variability. One thing to note is that when you're doing these analyses, and you see this in many papers, you get these really big blocks of data. So for example, in this case, we have a number of SP factors. We have another group here, CEBP proteins, RUNX proteins. You see a big group of them with very similar scores. And a lot of times people want to go and look at that motif and say, okay, well, GAB, this one here is the gene I'm going to go after. RUNX1 is the one I'm going to go after. Remember that motifs across transcription factor families have a lot of similarity. And so just by going and finding that one motif is the one that you want to go after, that's not enough information. You just know that you have the transcription factor family, and you need to get more information about what exactly that transcription factor is. So unfortunately, ArchR right now, we like to call it in the lab binning, but you could think about transcription factor grouping. Ideally, you would plot these plots where you actually compress this into a motif of that's somewhat similar. So in our case, we typically use 70 to 80% similarity, and then we generally call that a transcription factor, a family that then we can move forward with and further understand. So here's an example with the FOS transcription factors. You see that if you look carefully, there's some differences in the size of the letters, but you have to remember how these were determined. It's typically assays in different cell types. These are very context specific. So we would just call that the FOS transcription factor. And it's always a good idea to look at the tracks for attack sequencing data, because you want to make sure that when you're doing these analyses that you're actually reading out meaningful information. So for example, here we know that there's a transcription factor called CEBP alpha in lung cancer that's active in these states here. I'm not going to go through the details of how we figured these out, but just suffice it to say that these over here are sort of earlier stage cells, and then we have later stage cells. And you can see a very clear chromatin change there, and then that mimics what we also saw from the motifs. So just to give you another example of how this works, what we basically did is we could use those motifs, we could find where motifs were changing. So this is the same map I showed you before. These are the early cells, and then these are sort of later cells, and these are metastatic cells. And we could find motifs that correlated with those, and we could do further analyses to learn about the biology of those clusters. And we could do other approaches, which I'll mention in a moment, to find out exactly which transcription factor was driving the disease. So for example, we were able to find from these sorts of analyses that RUNX2 was a very important transcription factor in this transition from late stage disease to metastasis. And we actually found that there were rare cells, primary tumor cells, that looked very similar to metastases. And then we could go back and do imaging analyses, and we found that these RUNX high cells that we found from chromatin accessibility data were these rare populations of cells in the tumors that correlated with loss of their identity, as well as the activation of other genes associated with the transition to a mesenchymal state. So these, the most powerful way these tools are used is making hypotheses and then going and testing these hypotheses computationally or helpfully functionally. And then we were also able to confirm that this RUNX2 gene was very important in metastasis. So when we ablated it, we could see that there was reduced metastasis as well. So the way that we got to RUNX2 as a transcription factor as being important is instead of just looking at the motif, we then went in and said, where does the motif, how does that motif change, correlate with changes at individual loci? So this approach is commonly called gene scoring, gene activity scoring. And I showed you the way that we did this before with the transcription start site and this decay function. But the ArchR tutorial today does some different things where it does some blocking. It doesn't really use the same complete exponential function, but kind of weights the gene area a little bit more. But the main, what we get from these sorts of analyses are enriched or information about chromatin accessibility at a gene of interest. And so you can go then and you can, instead of looking at a motif, you can then go and look in your data, in this case, the example of the UMAP I showed before, and start identifying clusters. Because that can be really the hardest part of working with single cell data is figuring out what you have and what you want to learn from those data. So in this case, for example, we had CD45, which is a known immune gene, or CD19, which is associated with B cells, vimentin. We could then go and take that chromatin accessibility information from the loci itself and then see where those clusters light up. And so then we could say, okay, well, these are likely CD45 positive cells. These are likely our B cells. These are likely vimentin high cells. These are our metastatic tumor cells. So you can start to make these inferences as well. Again, a point I'd like to suggest is that every time you're sort of looking at these gene scoring to make determinations about cell populations, always go back to the tracks and make sure that the tracks are telling you what you think that you're seeing from your data. Because if it doesn't look right, either something's off with the analysis, or it could just be a complicated gene that has some regulation that's maybe messing up these standard approaches. But for the most part, they work quite well. You can also use tools like ArchR to go in and look at individual peaks. I don't typically recommend looking at the peaks in this way, but it can be helpful because you can find specific sites. You can do the standard peak calling analyses and find changes in your data set, features that go up or down. But in single cell data, this is much sparser than bulk data. So there's a lot of noise in doing these sorts of analyses. And then again, you can actually use the ArchR platform to go and look for changes at specific loci. So in this case, you see that this gene, this erythroid gene, you see more peaks at that site, which would make sense why you see those changes. I also want to mention that something that we do a lot in the lab is reference mapping. It can be powerful to make use of the data sets that have already been generated. And don't limit yourself to ATAC sequencing data sets, because the nice thing about this gene scoring approach is you can actually similarly use RNA sequencing data sets for reference mapping. And for those of you who aren't familiar with the term reference mapping, it's really just taking someone else's data sets and looking for cells that are most like your cells in your data set and can help you identify those cells. So I'm just showing you an example of how we did this in our case, where we took a normal lung data set and then could find things that we knew, where the AT2 cells were, et cetera. But it can also be important to note that if you do this reference mapping on cells like if you see here, the tumor cells are all gray. The reason they're all gray is because when you do the reference mapping with the data set that something's not that much like it, it will call it something. So again, every time you're using tools, understand the limitations of them, understand what they can and cannot tell you, but they cannot help you identify a new population if it's not already in that data set. And so what you can also do is you can take these signatures either from motifs, either from gene scores, and then you can do signature-based analyses. So here's an example that we did with lung cancer cells, trying to see if the cells look more like a cell type, an AT1 cell type, or an AT2 or an AT1 cell type. And so we could basically show that these signatures would associate more or less with specific populations. Here's a Pearson correlation coefficient, where you see that the more AT2-like cells are in this population, the more AT1-like cells are in this population. So you can also use other signatures that you find both ATAC and RNA data sets to make correlations and inferences from your data. And you can also do, in this case, with these cell populations, we had built those signatures from doing the motif analysis and gene scoring analysis that I talked about before. I also wanted to mention my favorite analysis of these sorts of using these data sets, which can help you identify function of transcription factors, which can allow you to infer if a transcription factor is behaving like an activating transcription factor or like a repressive transcription factor. And this takes into account what is happening at the motif itself. I think I'll go one slide ahead and go back. So what's happening at the motif itself versus what's happening at the gene score, at the locus itself. And if you see a correlation, it's likely that this motif and associated factors binding this motif are activating gene expression. So in this case, this is an example of this transcription factor 1 cut 2, which we found was important in late stages of disease and metastasis. And so if we looked at the gene activity score, we saw that it lit up in the same regions that you saw the motif becoming more open. So opening of that motif was associated with more of the loci becoming more open. When you look at ZEB1, which is a known repressor involved in EMT, you can see that the gene score becomes higher and the motif score becomes lower. So that means that the ZEB1 locus becoming more open is leading to genome-wide suppression of the ZEB1 motif. So we actually did this across many different data sets, as well as in this case, across these specific populations of cells we identified in lung tumors. And it's given us this atlas that's been incredibly helpful for us to find new proteins that are regulating genes in lung adenocarcinoma. And so in my last few minutes, I just want to mention a few other interesting things about ATAC sequencing, maybe not quite ready for single cell, but can be used, is that you can actually find regulatory units. Oh. Okay, so we're either lagging or
It looks like it's caught up now.
hopefully there's not a internet thing going on.
Are you trying to?
Yep, okay, that's working. Maybe I can just talk until, let's see, I don't know.
I don't know.
Yeah. Okay, well, I'll tell you what, and hopefully the slides will catch up. So another thing you can find is that, remember, you have DNA information. So with DNA, you can actually find sequence variants in open regions of chromatin. So people have shown that you can essentially find mutations from these data and make correlations. And now with multi-omic approaches, you can actually do ATAC and whole genome sequencing on the 10x platform together. The other thing that is useful is transcription factor footprinting, which we talked a little bit about, and won't be much in the tutorial today, but this is becoming really powerful, and people are using machine learning to tease this apart. So let's see, we're not advancing there, but I think I'll just conclude since we aren't having the slides advance, but Anthony has plenty of more detailed information in the vignette, which I think will be very helpful. And I'm also happy to take any questions that you might have. Any burning questions? Yes.
Going back to, say, bulk ATAC-seq versus single cell ATAC-seq, what kind of coverage loss do you see in single cell ATAC versus doing bulk?
I mean, I don't know if I have a good number for you, but it's really going to be your sequencing depth. So it's quite substantial. Like if you kind of think about getting 1 or 2 reads per enhancer, if you go and you look into a bulk data set, you're going to have many, many more of those. So the scale is quite different, but probably it depends also on the enhancer region itself, how much coverage you get there. But yes, it is quite a reduction, but that's why it's powerful when you can aggregate that information and to the motifs, because then you get more signal than just looking at a site itself. What's that? So yeah, footprints with the sparse data. Yeah, I think it is, and the slides I wasn't able to walk through. The more data you have, the better for that. So either aggregating pools of cells can be helpful, and now people are using machine learning to actually really scale this and figure this out. But footprinting can be pretty hard without, like, on a cell-by-cell basis. All right, I think let's maybe take a moment to a few minute break to switch over, or do you want to keep going right through?
Yes.
Okay, yeah.
Maybe a 5-minute break? Can I talk to the mic?
Yeah.
Thank you so much, Dr. LaFave. Let's give a round of applause, please. Sorry about the technical difficulties. That was really good. Okay, so we'll take a 5-minute break. People can grab some lunch or go to the bathroom, and we'll sit up here. So yeah, 5-minute break.
I didn't see your text message, Alex.
Yeah, no worries.
Sorry.
Yeah, if you could maybe just stick around like Clista's done and help
Yeah.
with the Q&A and stuff, that'd be great. Yeah, I wonder what happened with those slides. It's like it was working.
You were like too
Zoom? No, I think. Oh, maybe. Ooh, could be. It's still actually showing.
They look blurry too. Yeah, on there at least, but not on Zoom.
I was wondering if it was the internet, but then.
No, see, it's still showing. Yeah, that's weird.
Oh, it's still showing the slide.
It's still
Yeah.
showing that same slide. Test. You wanna get set up with me? Yeah.
So not like.
Yeah, I got it. Okay. Yeah, we'll do it. Thank you very much. That was really.
I hope that.
Ask you some questions. Thank you very much. Yeah, thank you.
I'm sorry, I didn't see your
No worries,
text
no worries.
message.
No, no.
I didn't think about moderating the questions.
I can do it up there.
Loud. Right? I'm a loudspeaker.
What? Say that we're good. We're fine. Okay. Yeah. I'm a little worried about this screen situation. I'm gonna try to share to Zoom. It should cast to both, although now I'm a bit worried about that, but let's see. Actually go to those. So if you could make a quick announcement at the start. I guess two things. First, that we might go a little bit past 2 since we started a bit late, of course, before. And secondly, people should just download that HTML file from the Net website. That's where we're gonna follow along. Okay, cool. Is that on Zoom?
Oh, it's on Zoom, but then this doesn't share automatically.
There might be some kind of issue with this, unfortunately. Okay, maybe what we could do. Yeah, try to just plug in. Are you plugged in there? Okay, that sucks. Let's see.
Oh, it's out.
It seems like it's super delayed.
Yeah.
So can you try scrolling, I guess, or?
Okay, it's a little laggy.
Okay. I guess we could just stick with this for now. And can you try scrolling through the whole thing and just see maybe when you get to like a UMAP? It's actually a good point. I guess there's a lot of like
Just
render.
froze.
Yeah. It just froze just now.
Yeah.
Like on your computer? Okay.
What was the other option that you guys had?
What we were doing before is essentially not using the Zoom in the room,
Yeah.
and just you would cast, like you would like share to the room and to Zoom at the same time.
What do you think that? Should we try that instead?
Okay, we could try that. Yeah, we could try that.
Alright.
So in that case, let's see how we do that. So you would essentially need to share to the screen. So I think the way we do that.
Oh, do we have to log out of Zoom first? Yeah. Just to avoid like the connection issue.
So can you try plugging in your laptop cable? It's doing something.
Well, it was like a, or like, is it keep on lagging because of it being connected to Zoom?
Yeah.
Oh.
You know what I mean?
I think
we
it's
can
'cause you're
disconnect
doing it
from
over
this.
wireless to
Okay.
the.
Okay, now it seems to be. Are you sharing it?
Yeah. And it's still lagging. Let me try.
When you say lagging, your computer's frozen?
Oh, your computer's
Right?
frozen?
We call here, right? Okay, guys, we're gonna get started in just a few minutes.
There you go.
Okay, let me make that announcement again. So we're probably going to go a little bit over in time since we started behind. So the workshop will probably go a few minutes late. The other announcement is to make sure that everybody has the HTML file downloaded from the Vignette website. The link was sent out on the previous mass email to all the RSVPs earlier today. So at that, I'm going to go ahead and introduce our next speaker, who's going to be guiding us through this single cell ATACseq workshop. His name is Anthony Griffen. He's a third-year graduate student in Dr. Lindsay LaFave's lab. Anthony graduated from San Diego State University in 2020 with 2 bachelor's degrees in biology and computer science. His first research experience as an undergraduate was with Dr. Scott Kelly, developing a computational pipeline to analyze environmental microbiology data sets. He then came to Einstein, hoping to continue his developing skills as a wet lab biologist and bioinformatician. He is a CMBG T32 training grant recipient, is now working on his primary doctoral work. He has been able to continue expanding on these skill sets, investigating biomechanical regulation of chromatin states in lung cancer. Outside his research, Anthony enjoys trying new restaurants and is always looking for new recommendations. Take it away.
Okie dokie. So I'll be going over the vignette. So unfortunately, this won't be a live demonstration. I'll just be going over code and a vignette that I had run previously. But of course, all of the code that's in here should, in fact, be compatible in the event that you want to repeat it on the HPC or on your own device later on. Some of the images are a little bit blown up, but of course, I'll zoom in and out, and we'll adjust that later on the GitHub link. But basically, as Lindsay mentioned, there are dozens of tools to work, or at least a few to work with single cell attack sequencing data. Oh yeah, huh? I could just hide it, right? Usually there is, maybe not. All right, at the very least. Yeah, that's enough out of the way, right? Yeah, all right. So today we'll be focusing on a tool called ArchR, which is published and maintained by the William Greenleaf Group, and they were part of the original team that had discovered and optimized the attack sequencing method. So I find this tool to be probably the most straightforward, especially in reading the documentation. It's rather comprehensive. So I encourage any, even outside of the vignette, outside of the specific methods I decided to focus on, they have a lot of other tools, a lot of other analyses that you're capable of running. So feel free. And then even a lot of the descriptions I borrowed from their tutorial as well, given that I just couldn't say any better than they had pretty specified. But basically, this is a brief overview as what Lindsay had described. One thing to note is that they have this specific file format called arrow files, of which I'll get into the details of which shortly, which, like Lindsay mentioned, might make it a little bit more difficult to work with other tools, but there are workarounds since this tool was initially published in 2021 that make it compatible with other attack sequencing technology, so of course, or analytical tools. So of course, keep that in mind that this is not the end all necessarily either. You can use scATACseq. There's other tools that are better at performing certain tasks, but keep that in mind. Now, they obviously did not update this recently, given that there's now a second iteration of scATACseq called scATACseq, which they basically tried to specify precisely what RCHAR is capable of that these other tools can't and why to use RCHAR. But of course, scATACseq tried to rectify some of those differences in the latest version. But of course, like I had said, I still think RCHAR tends to be the most complete. But really quickly too, in case anyone wants to work on their own device in the event that they have R already installed, I just have a few notes on how to install the necessary packages. You also need to install Max2, which requires Python, so it might be a little difficult, but for those who might be more computationally comfortable, feel free to just work on your own device. The tutorial data set is small enough to work with that it should work on your own device, assuming that you have a few gigabytes of storage left, which I hope so. So the first thing, of course, this is all the following code is in R. The first thing to do is to load all the necessary R packages. So of course, loading an RCHAR and this other display package. And of course, that's just returned. They have this nice little Archer image there every time you were to run it. And the first thing you want to do is read in the tutorial data set. So as I've written it here, this is only a subset of a tutorial data set that RCHAR has included. It's a hematopoietic data set. So I'm using the sample that specifically purified cells from bone marrow mononuclear cells, the cell population. But there are two other samples you can pull from in the event that you're comfortable with working with a larger data set. But for our case, we're working with a single fragment file. So you can download the complete data set with the following command, but otherwise we'll deposit all the necessary information you need on that GitHub link, including this fragment file. And for those who are unfamiliar, a fragment file, as specified here on the 10x Genomics website, is basically containing the start and end genomic coordinates of all aligned sequence fragments. It's the direct result of using another tool called Cell Ranger. Next week, when Alex talks about single-cell RNA sequencing, he'll review a little bit of how to use Cell Ranger, so how to work from raw sequencing files to generate these fragment files. And it's remarkably similar with attack sequencing data as well. The main difference is instead of using Cell Ranger, use Cell Ranger Attack. But I've included the link here in case anyone's interested. They have another tutorial data set to work with as well. I think it's also hematopoietic, as people tend to like to use those in their vignettes. But without further ado, so the next thing you want to do, just to begin with, is to specify the reference genome by which you've generated your fragment files. So this is depending on the species, the sample you're using. This sample specifically was using the Human Genome Reference Build 19. But of course, this RCHAR platform supports HG38, MM10, and other reference genomes. And even allows you to generate your own reference genome in the event you're working with different species, zebrafish, et cetera. Might prove to be difficult, but they have some compatibility there. The next thing to do is, which I've commented out just for the sake of simplicity, in case you just work with the normal computational resources that the HPC allots you on the cluster. But RCHAR is also capable of multi-threading for those who are familiar. So basically, you can assign multiple threads if you have multiple CPUs. That will enable some of these functions to work in parallel. So instead of one CPU tackling this function, this problem, you're having multiple, so it runs quicker. So just keep that in mind in case you're curious. Then finally, the first thing we want to do is generate these arrow files. So from our fragment files. And as you might have noticed, previously, I had specified the variable, the input files to refer to the file path of where this fragments file is located. And then I've also specified as another parameter to this particular function, the name that I want to have assigned to this sample. And of course, you might have a list of names if you have multiple samples, multiple fragment files you're working with. The next thing to note is this command will also perform those quality control that Lindsay had described in her presentation. I'm working with mostly the default parameters that ArchR has set, so they're rather loose. You can always, I recommend probably starting with these values first, and then in the event that you feel as if you're accidentally including cells that are of poor quality, you can change these parameters as to how you see fit. And I'll go over briefly in a second of what those filtering steps look like. The other things to note is that there's a tile matrix that's generated, which I'll talk about a little bit later, which basically aligns these fragments in these 500 base pair uniform length bins, and then a gene score matrix, which of course Lindsay had mentioned gene scores, but that's automatically generated at the beginning as soon as you generate these arrow files. So just scrolling down, oh, there we go. Looking at some of the QC plots that are generated. So when you run, there is a file that's, or a folder that's automatically generated called quality control for each sample that you may have. So some of the quality control metrics include the unique number of nuclear fragments, so any fragments that are not assigned to mitochondrial DNA. So of course, if you have a low number of fragments for a given cell, that implies maybe the cell is damaged or poor quality, so you'd like to discard any of those. In our case, since the filter is rather loose, any cell needs, as specified up above, 1,000 fragments, that no cells were discarded from that particular filter. The other thing is the transcription start site enrichment score, which Lindsay described, which once again is talking about how there's an expectation that a lot of reads will be aligned around gene transcription start sites. And in the event that's not true, that might also imply a cell of poor quality. So this score is basically the ratio of reads that are aligned along those transcription start sites compared to those flanking regions. But and you see in that case, some cells were excluded with that given filter. And after accounting for these two quality control metrics, you're left with around 5,000 cells left, which might be a rather small data set, but like I said, just for the sake of this tutorial, we didn't want to make it too computationally taxing. And then this is kind of a spot check QC metric. Lindsay also described this one, looking at the fragment size distribution. So you have some expectation that you'll have an enrichment of reads that will be either smaller or larger than the amount of DNA that's wrapped around a nucleosome. So for instance, you see an enrichment of reads that are around maybe 50 to 100 base pairs in length. Those are basically insertion or fragments that you've caught that are in the intervening sequences between nucleosomes. And then of course, in the event that you capture a portion of the genome that's including a nucleosome, you'll expect to have at least 147 base pairs, but of course you'll have a little bit more with any other intervening sequences that are captured. So you see an enrichment of reads or fragments around the 200 base pairs in length as well. So in the event that you don't capture that nucleosomal periodicity pattern that's expected with ATAC sequencing, that also might say something about the sample quality and might encourage you to instead discard a sample, especially if you're working with a high number of cells. So some of that's described here in the notes, so feel free to refer back to those in the event or when you run through this yourselves. So one thing that was kind of novel about ArchR at the time was that there weren't a lot of ways to predict doublets. So for those that aren't familiar with a lot of these 10x droplet-based sequencing approaches, there's a likelihood that you might accidentally encase more than one cell in a droplet. And in effect, you might be getting a number of reads that are, basically accounting for more than one cell. So you want to make sure that, albeit the data might interpret it as one cell, the fact that it is two makes you, you want to discard those cells basically, or those doublets from your analysis so it doesn't skew the results later on. And the way that ArchR does it is they synthesize these in silico doublets. So these are all computationally derived and determined by just mixing the reads from thousands of different combinations of individual cells of your data set. So of course, there's an expectation that a doublet will be some combination of multiple cell types that you've captured. And then you can basically project these doublets into this UMAP embedding. So UMAP being this dimension reduction technique that I'll describe more in detail later on, and identify whether or not your cells or your samples cluster near these synthetic doublets. And if they do, then they're likely doublets as well. It's kind of the idea. Similar approaches are adopted for single-cell RNA sequencing as well. This whole idea of computationally determining synthetic doublets and comparing back to what's in your data set. So that's the more simple interpretation of how this works. But you can also modify some parameters, including how many cells you want to count near a pseudo doublet or a synthetic doublet to consider, all of which might influence how many the doublet enrichment score and how many cells you might end up discarding later on. So, sorry, these images are a little offset, but these are examples of some of the doublet score plots that are from our sample. So this is also deposited in the quality control folder automatically after you run this command. And of course, you can see that these are basically different ways to visualize the enrichment of a doublet score. And you'll see these unique clusters that are more likely, more predicted to be a doublet. So of course, you end up discarding those. And I'll describe that step when those cells are ultimately discarded. But for now, we've just added those doublet scores to control later on. And then finally, this command, the rchart project command, allows us to group multiple arrow files together. So in our case, once again, we have a single arrow file, but you can imagine another context in which you're working with multiple samples. So what I do is that in that create arrow files command, I basically captured the arrow file that's assigned to my sample that I'm working with, and I'm going to deposit all this information in a directory titled theme tutorial. So it's important when you see these output directory parameters that unless you specify a certain path, a file path in your computer, these directories will be deposited wherever you're working in your computer, basically. So, and I've also included a command down below, which works very similarly, in which the save rchar project function, where you specify the name of the project. So of course, in running this command, it's called project1, and then the output directory you can name separately as well. And you can choose whether or not to include the arrow files. Granted, I will recommend that you do, given that you can't load the project back in if you were to exclude them. So there is an option in both the save rchar project function and in this one to exclude those arrow files because they ultimately take up space sometimes. But of course, that's only in the context of you want to save a version of it that has a series of plots you want to refer back to, but don't ever want to analyze or use again. So just keep that in mind. But the other thing, the next step is basically to filter those doublets. So this is based on that prior command in which it's made that doublet enrichment score. But the maybe more interesting parameter here is the filter ratio. So basically this parameter is trying to make sure that you apply this consistent filter across multiple samples. So you want to basically have a certain ratio of cells that end up being discarded just in case certain samples were run with different cell loading concentrations, because you can imagine some samples might be more dense. But either way, so in other words, the higher the filter ratio, the greater the number of cells that are potentially removed as doublets. And they have an example of an equation here in which that's the case, but the default ends up being around like 5% of cells can be labeled as doublets. After running this command, if I scroll down in the output, you can see that that is in fact the case. It removed 243 cells of our nearly 5,000 because they were too similar to the synthetic doublets that we've determined. And then I saved the project again. So this is basically a version of the project with the doublets included and one without. And then you can load it back in. This is just an example of loading a version of the project, whatever iteration or version that you're currently working with. So that just makes it easier to refer back to versions of the project as you've left it. And then finally, in talking about dimensionality reduction, this is basically a method to work from a high dimensional space to project onto a two-dimensional space instead. So of course, with sequencing data, you have for a given cell, a lot of information. You have a number of fragments that can be assigned to different peaks. And the main issue, of course, which Lindsay also referred to, is the sparsity of single cell attack sequencing data. So I'm going to make a parallel to single cell RNA-seq. I know it's next week, but the main idea with single cell RNA-seq is that you end up identifying highly variable genes in which you can begin to consolidate your data into lower dimensional space. But with single cell attack-seq data, the data is incredibly sparse. So you can imagine that it's almost closer to binary. So of course, Lindsay showed an example of a matrix in which it's somewhere between 1 to 3 in the values that are assigned to a given peak. So it's not a lot necessarily. And the many accessible regions are not ultimately transposed. So you have a lot of zeros in this matrix. So if you're to do a PCA or just any kind of dimension reduction from the beginning without any more processing, you could see that, and you end up having a lot of intercell similarities. So you're not necessarily able to parse out differences as well. So you have to do something else. And this is what ArchR does specifically. Other tools have adopted similar techniques, but they use this iterative latent semantic indexing procedure. It's kind of adopted from a natural language processing tool. But the main idea is that I don't want to go too far into detail or too much in the weeds, but it's a tool that enables you to identify peaks or regions that are more unique to a sample. So this method is also further strengthened through multiple iterations. So the first one, you basically look at the peaks and identify the most accessible features. And then the second iteration, you try to identify the most variable features from that subset. Basically from that data, you can then begin to identify clusters, cells that share a similar profile using a nearest neighbor technique instead. So then you end up saving that final project. But I will specify too, I've also let in room here for you to mess with those parameters, because here the default is just 2 iterations. So you have 1 iteration to identify the most accessible features, the second to identify the most variable. But of course, you can continue to iterate to eventually get to a small, especially if you have a rather large data set, you might want to iterate a few more times. And also to enable, so it's not doesn't take too long to process. You can also, end up working from a subset of your total data set. So in this case, they're using 10,000 cells, up to 10,000 cells. Of course, in our case, we're working with a rather small version of the data set, but you can imagine this might be important in other contexts in which you're working with a lot more cells. And then you also have some limitations on how many variable features to consider and so forth. So you feel free to mess with those parameters and see how that ultimately changes how your cells end up clustering. And of course, like I said, I left space here, and that is an option in which, and also ends up being very specific to your own data sets. So of course, I definitely encourage to continue modifying and to continue looking at what might be different. And then here is actually where it ends up being very similar to single cell RNA sequencing. In fact, it uses Seurat, which is a common platform for single cell RNA sequencing, and identifies clusters. So once again, this is using that previous algorithm, the iterative LSI procedure, and then finding these cells that have similar profiles, and then basically cluster them together, depending on how close they are in this low dimensional space. So of course, modifying the resolution will determine how many clusters you ultimately derive from your data set. I left as the default value of 0.8 as well, but feel free to modify that particular parameter depending on if you want more resolution, meaning higher number of clusters, if you anticipate that there are cell types that are rather small subsets of your total data set that might be worth doing. And of course, the other way might be helpful too. So then lastly, using the iterative LSI, reduced dimensional embedding, you can then make a UMAP. So UMAP is another approach which I feel like most have probably seen these UMAPs before. We just saw in Lindsay's presentation. Now, Lindsay also alluded that there is some degree of controversy of using them, but especially if you visit Twitter, there's a pretty active discussion about how useful they are. But I think in the meantime, they're rather useful for maybe the broad strokes of looking at where the different clusters, the different cell types of your data set. But of course, it is always to take into consideration that you ultimately have to back it up with other descriptive characteristics of these cells, whether it be certain gene scores or gene activity that delineate precisely what they are rather than where they belong on the UMAP instead. But so this is another option where you can mess with some parameters with UMAP. So for instance, you can change the number of neighbors used to construct this. So this, in modifying this, you can basically specify whether or not you focus on local versus global structure. So if you want to look at basically low values will push more on local structures. So if you don't consider as many neighbors that are adjacent to it, then it tries to preserve those relationships instead. And of course, the other is also true. It goes the opposite direction. And then the minimum distance parameter here also determines how tightly the UMAP is allowed to pack points together. So 0.5 is the default, but if you want tighter clustering of your data set, this is another parameter you can modify. And of course, a lot of these are just, you test them, you see how they work, and you just work with what you have. And I can't necessarily recommend one way or another. The default's always a good place to start, but of course, that's something you learn along the way. Then finally, you can plot your UMAP embeddings in a few different ways, and you can label your cells. I labeled it here by sample, but of course, this would probably be more informative if you have multiple samples. And then I've also labeled by the clusters that we've determined in those previous steps. So if I scroll down, you can see the first image is basically the sample, which of course is, once again, we only have one sample, so they're all the same color. And then finally, the clusters. So we've identified a total of 7 clusters of our data set. But yes. You can identify clusters of your data set, and then you begin to make inferences precisely what these cell types might be. But there's a few other things you want to do to basically begin to assign those cell identities to each individual cluster. So this is something that Lindsay also spoke to. So there's something you can do with the ATAC sequencing data. You can basically adapt these gene scores. It's basically a way to estimate gene expression from chromatin accessibility data. So ArchR does it a specific way. They have model 42, they call it. So obviously, there's been different ways to do it that have been adapted. But their way basically tries to take a few things into consideration that maybe weren't taken into consideration before. So they want to take the idea that accessibility within the entire gene body contributes to the gene score. So in other words, not only the transcription start site, but they also wanted to elongate to include a stronger score if it starts to project a little bit further into the gene body. So that's what that first parameter is referring to. They have this exponential weighting function, which is true of this first model as well, that basically tries to progressively determine the activity of other distal regulatory elements in a distant-dependent fashion. So, of course, the ones that are closest to this first gene body spanning area are still given some degree of weight, but then that's rapidly dropped off. And then finally, they also want to impose some gene boundaries. So you can imagine that parts of the genome are rather busy. They have genes that are in close proximity to one another. So some peaks, although you want to make sure that they're not given value or used to attribute to a certain gene score when in fact it's regulating a neighboring gene instead. So they basically remove these genes that intersect are basically not counted, these peaks that are instead maybe likely attributed to this other gene. So that's the way that ArchR does it. There's been a few benchmarking papers that try to compare the two, but these two methods are pretty robust. And I'll show you some examples of what that might look like. So some marker genes. I just did some examples of some populations that we anticipated to have in our data set. So some CD34 to mark early progenitor cells and these other markers here. This plot embedding function can be used to use the gene score matrix. So once again, when you created the arrow files, that gene score matrix was already included. So those values have already been assigned to each individual cell. And then in this name parameter, you can basically assign whatever genes you're interested in the list. So I've basically made a list here, and you can change, you can add any other genes of interest. And then, of course, I want to use the UMAP embedding that we've adapted. And then this impute weights function I'll return to later on. But just to begin with some examples of these marker plots. And although you can see some examples of a lot of cells that light up, in gene scores for this individual marker. And that's true of PAX5, maybe more true here. But of course, it seems to vary depending on what marker you're looking at. Now, one thing obviously is that these can be made maybe a little bit prettier, given that some cells, for instance, light up here in this gene, but the neighboring cells don't. But of course, you can maybe make conclusions that this cell population, even if some cells didn't happen to capture those transposition events, likely still are have high expression of that gene. So what I mean by that, the way that can be rectified or fixed, or maybe just modified for visualization purposes, is you can add impute weights. So what ArchR does, it uses this function called magic. Once again, something that is sometimes used in single-cell RNA sequencing as well, in which you then begin to also, it's like this approach, it's another graph-based approach where any genes that, or any cells that are near others that have a high gene score for some marker gene, if it's in close proximity to a number of cells that have that high gene score, then it's also given a more weighted gene score that's more similar to those. So this might be more easier to interpret if you begin to see how these plots look. So for instance, before there were a number of cells that were highly enriched in CD34 gene scores. And of course, if you were to begin to weight them so that multiple of its neighbors share that same profile, then it's more easy to parse out, okay, this cluster is high in this particular gene, and so on. So that's true of all of these. But once again, as Lindsay mentioned, because you begin to bias some of your results, it's always important to look back at the chromatin tracks and to look at whether or not you see peaks assigned to these given markers independent of some of these visualization tools. But basically going through the list of all those markers that I'd specified. And you can see they also rather uniquely represent different clusters. So of course, that's also reaffirming that the clustering approach you're using does in fact capture specific cell types. Now, one other thing that we kind of already referenced as well is that single cell ATACseq data sets are essentially binary. So it's often difficult to make broad conclusions about some of your clusters and what how they might relate to the other cells of your data set. So you end up making pseudo-bulk replicates. And what I mean by that, a pseudo-bulk basically refers to a grouping of cells to a single pseudo-bulk data set. So it's kind of mimicking almost like bulk attack sequencing instead. And you can group by a number of things. You can group by metadata that you maybe have added, ways that you've classified your cells. For our purposes today, I'm just grouping by clusters. So of course, every cluster, instead of each per cell basis, you can then begin to make inferences generally of how these clusters, their chromatin accessibility profiles compare to others. Now, of course, the main consideration is that in this case, you're basically making the argument that, or the assumption that the single cells that are being grouped together, in this case, cells that are in the same cluster, are sufficiently similar to one another that you don't necessarily care to understand the differences between them for any analyses in which you use these bulk replicates. So because some of these steps take a considerable amount of time, I've also saved different versions of the ArchR project along the way. So of course, you can pick up the tutorial from after I've saved this project, because I'll deposit the intermediate steps as well, the intermediate projects, to make it a little bit easier. And of course, you can see that some of these steps range in how long they take. For instance, this one takes a number of minutes. Some of the earlier steps were rather fast. Any of the plotting of the UMAPs and so forth are rather computationally quick to do. And I've also shown another example in which you can load your project back in. Okay. So the next step that we're going to do here is calling peaks. So likely you guys are a little bit familiar from previous Omics Club vignettes, but what we want to do is call peaks, but because it might be difficult to do on a per cell basis, instead we want to use it with our replicates or bulks, pseudo bulk replicates instead. So a few ways that ArchR does it that maybe others don't. They weighed a few considerations with how different ways to do it. So for instance, you can have peaks be a fixed width or variable width. So you can have, you can imagine that every peak. So let me actually back up a little bit. So each cell has a certain number of fragments that then can be assigned a given peak. They can be a specific length, so 500 base pairs. So basically any fragment that falls in a particular range can be assigned to that peak. And then, but otherwise you can just also assign variable widths. So basically you end up combining multiple peaks but they make the argument that ends up making it a little bit difficult to normalize later on. So in their case, they're using 501 base pairs length. So just keep that in mind in the event that ends up being consideration for how you want to run other analyses. The other thing to note is they do this iterative overlap example. So in their documentation, they go into further detail if you're interested, but otherwise, they basically, if any peaks that they've called are overlapping, they first rank them by normalized significance. So the confidence in which you're able to call that peak, and basically any peaks that are given a higher significance value are prioritized. So any other peaks are discarded, and they do that a number of times over a number of iterations. And in effect, you end up not losing as many peaks that might have been lost if you were more harsh with how you remove any overlaps, basically. So this is where you end up using max2, which is a common peak calling tool. This is where you'll need to install it with Python, but hopefully the conda environment on the HPC will soon be updated to have this, and then you can run the following steps. But this function basically down here begins to do what I've described, adding this reproducible peak set. So these are across these reproducible replicates, and you group by clusters. So once again, this is basically assigning peaks that are specific to each cluster. And in our case, there are 7 clusters. So they also show an example of that max2 call that they're running behind the scenes. But, and this is just an example of where these peaks might be aligned. So they have, of course, it can range, and this is kind of almost more normal, that the peaks can align either exonic regions, intronic regions, etc. And in most of the case, of course, given how the genome is structured, there's a lower proportion of cells or of peaks that are assigned to the exonic regions. But this is almost more just a confirmation that at least your samples look normal or your clusters look normal. But maybe more interesting to adapt further analyses. First, we want to add this peak matrix. So this is basically the matrix counting the number of insertions that you have for each peak and assigning it to each cell. And then, of course, I also ran this get available matrices. So you can basically see that you have a gene score matrix now, a peak matrix, and that tile matrix I described at the very beginning, in which it's binned its fragments into 500 base pairs. But the peak matrix is likely more informative. Yeah, I think we should, because I'm going pretty fast, but. Yes.
I guess the input matrix of the raw data, it's like a matrix by number of genes by the number of cells. What is that? What is the raw input for a cell? Say you mentioned it was like binary. Instead of genes as the rows, like what is it as the rows?
Well, I guess the more common way to visualize it is by peak, for instance. So you have, I think Lindsay even showed it earlier. So this peak matrix, for instance, is one way to represent it, but you basically have for a given peak for every cell, whether or not how many insertion counts it has, how many fragments assigned to a given peak. That's the way to work with the data later on. Otherwise, the other ways to do it, of course, is the gene score matrix, in which you've kind of already made some inferences about, more similar to RNA in that case, in which you have a gene.
Is that some sort of peak calling algorithm or peak calling procedure to find those peaks?
Yeah, so that ends up spitting out a number of fragments. So that's like more, I guess, raw data in which, hoping I'm describing this right. I don't, is there, but you get a number of fragments, basically any number of lengths. So of course I showed that fragment size distribution, right? So you have a certain number of fragments that are maybe around 100 base pairs in length, but they range in size, and it's just anything that's captured between 2 TN5 insertion sites. So in that way, you can't necessarily, it's maybe a bit harder to work with that data directly. So basically you have, an individual TN5 fragment, but for a given cell. But of course, you want to do some more downstream analyses to develop these peak matrices, for instance, gene score matrices, to make it more interpretable. Good question. Any other questions about gene scores, about how R does it, that kind of thing? Okay. All right. Okay, okay. So we don't have too much more, so maybe I'm going pretty quick. But anyway, so the next step to do, so this is something that Lindsay had also referred to, looking at motifs. So here's an example of the FOSS motif, so this position weight matrix. So this is adapted or experimentally confirmed with a number of different protocols. There's a number of different consortiums, collections of these motifs that can be found online and ultimately used in a lot of these analyses. But here's one example in which it basically shows for a given nucleotide site the proportion of that site, the proportion at which are either this particular guanine, adenine, etc. And what you can do is basically see how accessible this motif is across the genome. And if it's more accessible, that might imply something about transcription factor activity. And these motifs can be either aligned across the transcription start site, in promoter regions, and regulatory elements. They can be in a number of places. But of course, the same pattern holds that this might also apply some cell type specific gene regulation. So of course, if you have, say, CP alpha, for instance, could be specific to a certain cell type, you'd expect to see its motif enriched in that population of cells. So in order to derive these motif scores for our data set, the first thing we want to do is we have our peak set. So we've developed that peak matrix of that 500 base pair in length peaks. And we also now want to develop basically a yes or no of whether or not a peak or a motif is in one of those peaks. So we're taking this motif set from scBP, which is one of those resources I described that has all these experimentally confirmed transcription factor motifs. They have a number of other options. They have ENCODE has another transcription factor binding site profiles that you can look at. And they also have the ability for you to look at the enrichment of motifs that are common from bulk scATACseq experiments and so on. So I definitely encourage for you to look specifically at this portion of their tutorial because they have some interesting approaches independent of just looking at scBP derived motifs. But regardless, in our case, basically what this does is that scBP has a number of motifs like FOSS, like CP alpha, whatever your favorite transcription factor is. And then we'll go through your peak matrix and go, is that motif yes or no in that given peak? And in doing so, then you begin to say, okay, if a cell has insertions in that peak and that peak is enriched in this motif, then that particular cell has a higher motif score. And basically the get marker features function does that, but of course on that pseudo bulk replicate scale, because it's a little bit more difficult to do on a per cell basis for now. But basically, then you begin to look at just differential enrichment of these motifs. So of course, it's not too similar or not too far off from just differential analyses you've run maybe in previous tutorials, differential expression analysis, etc. And you're basically trying to look at peaks later on that have a significance value that matches expectations and a log-fold change that's also interesting. So this basically function will derive the number of motifs that are specific to each cluster, in other words, or the transcription factors whose motifs are more accessible in each cluster. And you can specify how many motifs you would like to look at for a given cluster. in our case, I will say, I'm going to scroll down to the result from the visualization. Oh, sorry, let me repeat. So in our case, because we're only working with one sample, you can see that some of these clusters aren't necessarily enriched in a given motif, but you can imagine that if you increase the number of cells or have a more diverse data set, it might be easier to make inferences about which motifs are specifically enriched for a transcription factor. And we do run into that problem that Lindsay had described, in which you get some motifs that are members of the same family. So of course, a lot of these transcription factors, if they belong in the same family, their motifs are remarkably similar. So you have some phos motifs here, for instance. You have members of the SP family for cluster 6, and so on. So although RChart does not have this automatically in its functionality, there is ways to do it outside of RChar in which you bin them together based on their similarity. So you basically can maybe find more biologically meaningful results rather than getting a bunch of hits for basically the same motif or at least very similar motif. Okie doke. And then I saved a version of that RChar project just to make it easier to access later on. All right. So this is the last thing before the end of the vignette. So the last thing we want to do is, okay, so all those other analyses and looking at motif enrichment, we're all on our pseudobulk replicates, but we kind of want to be able to assess regulatory factors on an individual basis or individual cell basis. And the other weakness too, as Lindsay described too, is that there's an insertion sequence bias of the TN5 transposase. So in order to basically fix those issues, they use ChromeVar, which is another platform and can be used independently. It used a lot with bulk attack sequencing as well. And ChromeVar is basically designed for predicting the enrichment of transcription factor activity on this per cell basis. So the two primary outputs that ChromeVar returns is this deviation score, which basically looks at how far a given feature, or in our case, a motif, deviates from the expected accessibility based on the average of all of our cells of our data set. And then a z-score, which is basically looking at how far they deviate from the standard deviation across our samples. So they're very similar. They're kind of looking at the same thing, whether or not a motif is differentially accessible in a given cell. So the first thing we got to do is identify background peaks. So these background peaks, basically you want to exclude peaks that are more common across all cells of your data set. You don't want that to skew results one way or another. So you basically look at your peaks and assess them based on similarity and GC content and the number of fragments across all samples. So in doing so, you can then adapt whether or not it should be taken into consideration later on. And then from, and comparing it back to those background peaks, you can then make this deviations matrix. So basically for a given motif, you can see whether or not it's specifically enriched in peaks that are more specific to a cluster or a cell of interest. So then from that analysis, now that you have your deviation matrix, you can then look at certain transcription factors of interest. So of course I have GATA1, CP alpha, and so on, ones that we anticipate to have in our hematopoietic lineages. And then look at, I basically extrapolate features. So some of them have different annotations from CISBP, for instance, in which they have this numbering scheme. They might have multiple motifs that have been identified. So I basically pull those out and now have the annotations for either their z-scores or the deviation scores that we can look at out on our embeddings. So here, very similar to how we plotted our gene scores, but instead we're coloring by our motif matrix instead. And then the other thing is, of course, we specify which motifs we're interested in looking at. So of course, I'm just going to use all the marker motifs I previously specified here. And you could also use those same imputed weights here as well, so to improve visualization of your motifs. So here is how that looks in our data set. So you see enrichment of the CEP alpha motif here. And that's the case for these transcription factors across different cell clusters. So GATA one more specific here and so forth. So in combination with the gene scores and your motif analyses, you begin maybe more confidently labeling these clusters with specific cell types and are able to begin to maybe see some novelty in how they look in your particular data set. And of course, in this tutorial data set, they're just baseline hematopoietic cells, but of course, you can imagine that in the event that you're working with a oncogenic data set or some kind of perturbation, this could prove very interesting if you see a different distribution of these transcription factors in a same cell type or how they push it towards another cell type entirely and so forth. Now, this isn't necessarily, I ended the vignette here, so I saved the final version of that ArchR project, but of course, ArchR is capable of much more. So something that Lindsay alluded to too is that identification of transcription factor activators or repressors by comparing back to the gene score. So you want to look at the locus of that particular transcription factor, for instance, and see whether or not they're positively correlated or negatively correlated. They're positively correlated, likely it's a transcription factor activator, and the opposite is true if it's a repressor. They're negatively correlated and so forth. So you can do those analyses with ArchR as well as transcription factor footprinting and a few other techniques that might be useful depending on any hypotheses or questions that you may have with respect to your ATAC sequencing data set. But those are more the broad strokes. But of course, if anyone has any remaining questions, curiosities, stuff like that, please feel free to let me know because this is the end of the vignette.
Any questions? Maybe I have a question.
How big is the data set when you're getting from cell major? Usually you have those three matrices from regular cell major, from the single cell RNA.
Yeah. So the fragment file in this case for this tutorial data set, I think was like 150 megabytes. So it's rather small. But the arrow files end up being pretty large because they end up not only containing information on fragments, they also end up including the data on the gene scores, et cetera. All of that's kept in the single file format. So in that case, those end up being a little bit larger, gigabytes kind of thing. And some of my data sets, some of my ArchR projects, for instance, end up being like 50 gigabytes or maybe more. But it's completely dependent on how many cells, how deeply you've sequenced, et cetera. But this tutorial data set is small, but of course. Once again, it depends, but the tutorial data set, you can, assuming that you have, I don't think you'd need very much storage, you could probably do it on just your own device. So assuming you have RStudio, you have all the packages that you need to run the analysis. What I do, I mostly have worked on my own device, only up until recently as my data sets have gotten larger, have I resorted to using the HPC instead. But it's all dependent on how much data you're working with. And of course, the HPC makes that considerably easier if your computer doesn't have as much. I think storage is kind of the main limiting factor here. And the processing, some of these steps take a pretty long time sometimes. Maybe not some of these steps I've outlined here, but there's other things that ArchR does that may take up to 30 minutes if you're just using your own device, maybe up to an hour, depending on how much cells you have. So in that case, the HPC is kind of nice to use instead. So you're not just sitting there waiting.
Are there any methods of doing lineage tracing with single cell ATAC-seq?
There is. I've used them. ArchR has one. I don't really like ArchR's because it's kind of biased. You choose the start point and the end point. So I think in that way, it's like,
Yeah.
you have to have some. So you're kind of there's other tools. I've used another tool called Cicero, which is compatible with the ATAC-seq data. But in that way, then of course you have to transfer any work you've done in ArchR if you started there to using this other tool instead.
I see.
Yeah, but there are options. There are ways to do it.
I was wondering if you could explain like the reasoning behind the iteration process you talked about. Like, what is that actually used for, and why is it?
Like, for, with respect to like the load dimensions.
Variable features after that, like why does our chart do that? What benefit does that?
So it's similar. So for anyone that's worked with RNA sequencing data sets too, it's like, I think it's kind of analogous to like how you end up focusing on highly variable genes, for instance. And a lot of the times that a lot of the pre-processing you end up doing. But it's basically that because the data is so sparse, because ultimately like how many insertions you have for each individual peak, you have a high number of zeros, for instance, that in order to be able to distinguish one cell from another, you kind of need to consolidate the information to being the most highly variable features instead. So it's kind of trying to improve the ability to distinguish one cell from another by continuing to iterate through and finding more variable features. Because if you worked with the original matrices, for instance, I think ultimately they end up reading out as being too similar because they have so many zeros that are uncommon, for instance.
So you like find your accessible features first, and then among that subset, then you look at the variable features. That's sort of what you.
Yeah, at least that's how they've done it, yeah.
Okay.
And then you could continue doing that a number of times if, you feel as if you still haven't caught. Data sets can be quite complex depending how big they are too, so maybe more iterations are needed. Continue to dive down precisely what's the most variable thing. But
Thank you.
Any more questions? I think we'll wrap it up then. Thanks everybody for coming, and thank our presenters.